Hello everyone, welcome to a brand new video. This video will be a series on cellular respiration. It'll be a four-part video series. In this, we are going to focus on an introduction about what cellular respiration is. So as we go into that, what are we going to see in this video? I do want to begin talking about what catabolic reactions are, why they're important. The three main catabolic processes are involved here. We will talk about some other things that more in detail later, but like I have separate videos on fermentation, things like that. But we're going to talk about fermentation, briefly mention that these are all slightly different processes. And then we'll go over the overall equation for cellular respiration, discuss what redox reactions are, both reduction and oxidation. We'll discuss the electron carriers, NAD plus and FAD, and the role of dehydrogenase enzymes, and then I'll give an overview of the four metabolic pathways that are involved in cellular respiration proper. So as we get into this, what I want to start off with is just talk about what do we mean by the word catabolic. When we hear the word catabolic, what we are saying is we are talking about a type of metabolic reaction where things are broken down. Now, specifically, we are going to be focusing here that chemical compounds are going to be broken down. So let's just use a very simple example. Let's say I have a substance that I have called AB. And let's just say this is two chemical compounds bonded together, kind of like what you might find in disaccharides or something like that, like two glucose molecules. Now, and the chemistry occurs, we can break these apart. We can produce the substance separate A and the substance separately B. Now, when this happens, we also get energy release. Now, this energy that we are being released here is something like ATP, for example, or heat, light. But ATP will be our primary focus in this series. Now, what I want to really kind of think about is that this release of stored energy, we actually refer to this process as exergonic as well when we are looking at the release of the stored energy from the chemical bonds. Now, this energy that we release can now go on to do cell work, like transport work, mechanical work, chemical work, things like that. So this energy that we are released will go on to do cell work in the body. Now, there are a few different processes out there that are involved. There are three main ones that are referred to as catabolic processes that happen in living things. But these three, only one of them is really going to be, it's the second one that will actually be applicable to us. But the first one I want to talk about is called fermentation. Now, I already have video on fermentation where we talk about that fermentation is really a very partial breakdown. What is important about this is this deals in environments that are very low oxygen environments. So there will be very little oxygen or no oxygen. So you only partially break down substances. Okay. Now, I talked about that more in detail in my video on alcohol fermentation and lactic acid fermentation. Now, as we get into this, since it's low oxygen, we actually refer to this as being without oxygen, so it's anaerobic process. This is an anaerobic type of catabolic reaction. Now, whereas aerobic reactions, aerobic respiration, this one, aerobic respiration, which we also refer to as this one here, is cellular respiration. This is overall cellular respiration. Now, it does have to have oxygen. It requires O2. So it is aerobic, but we will see later on that it is that overall aerobic respiration, especially cellular respiration, does actually involve an anaerobic process. So when we're dealing with this, it is aerobic, and this guy uses organic fuels like carbohydrates as well. Now, with fermentation being anaerobic and aerobic respiration being aerobic, there is this very interesting one that is a lot like aerobic respiration and fermentation. It's kind of like a combination of the two. And it is called which when we put that in front of it, it means negative, aerobic. And this is anaerobic respiration, which is not the same as the previous two. What this guy basically is it is occurring with or without oxygen. So you can have O2 with or without. It doesn't care. Usually, most anaerobic respiration processes really don't care. Now, there are some that do. There are types of anaerobes out there, facultative anaerobes, obligate anaerobes, things like that. But if you're dealing with some anaerobic respiration, it can occur with or without oxygen as an overall process. Now, don't confuse that for individual organisms who can or cannot. But anaerobic respiration can occur without oxygen. I oftentimes just say it's oxygen agnostic as an overall process, but as we talk about that, we can see that it does occur without oxygen, doesn't need it at all. Oxygen is not needed. And so that means it must use something else. Like it might use, for example, SO4 2 minus, or maybe NO3 minus ions to actually act as an electron acceptor, sulfate and nitrate ions to act as an electron acceptor, not O2. Now, this will make more sense, though, as we get into the electron transport chain and actually discuss it. Now, one of the big things that I really want to focus on here is to help you guys understand how can we study cellular respiration as a process, because this is the breakdown of organic compounds. So the best way to understand it is to follow a chemical that can pass through all the processes. And glucose, as it stands, is really good at doing that. So if we looked at an equation for this to understand what cellular respiration is, we're going to use glucose. Glucose, as you guys should know, if you've seen any, if you've had enough biology to get here, you probably already know that glucose is a hexose sugar, so it is C6. Now, since it is a carbohydrate, it will be H12 because for every one carbon, there are two hydrogens, and there will be O6, C6H12O6, that this is glucose. Now, what we also have to do is have oxygen present to do this because this is an aerobic process. But it will require 6 oxygens to occur. Now, what we want to discuss further is when this happens, these 6 oxygens and a glucose will be scrambled. You will get rearranged, and we will form 6 carbon dioxides as a waste product, okay, from this. And as well, we will get 6 molecules of water out of that that are produced. I will show you how that happens in the last video. And 36 to 38 molecules of ATP that I can get out of that. And this is the ATP that can go on to do cell work. So this is our purpose of doing this, is to take the organic compounds, an organic compound like glucose in the presence of oxygen, to break it down and to do this. So this is why you breathe. You need to inhale to obtain oxygen to feed the chemistry that does this, and to exhale to remove the CO2 that is made as a waste product from that. Now, as I said, this is a scramble. And what we're doing when we're scrambling is we are going to take the electrons that are found in the glucose in the covalent bonds, specifically the carbon-hydrogen bonds, and we are going to nab those up, steal them, grab them up, and use those electrons to make some energy. So to do that, we need to learn about something called redox. Now, what is redox? Well, redox stands for two things. It stands for reduction and oxidation. So let's help you guys understand what redox is. To do that, we have to understand a fundamental thing about all atoms is that atoms are neutral. Why are they neutral? Well, atoms are neutral because they share the same number of protons and the same number of electrons. Now, because of this, the positives and the negatives equal each other, and when positives and negatives equal, they cancel each other out, and they have no charge or said to be neutral. So if we understand that all atoms are fundamentally neutral and have a charge of 0, we also know that you can add electrons or take electrons away, and this will affect the charge. We can make the charge go up to be more positive, or we can make the charges go down to become more negative. Now, how is it that we can do this? To make a charge go positive, we now have to have the number of protons outweigh the number of electrons. So we can remove electrons to make the charge go more positive. Or we can take electrons and add them in to make the charge go more negative, because now the negative electrons outweigh the number of positive protons. And this movement of electrons, either removing them or adding them, is the basis for redox reactions. Redox reactions, reduction and oxidation. So what we want to make sure you guys understand here is, before I actually get started, I just kind of like, as I explain these and give the definitions to these, there's a mnemonic I always give my students, and I always remind them of my pet lion named Leo. And Leo, the lion, we know that lions say grr. So Leo, loss of electrons is oxidation. So loss of electrons, oxidation. So what we have here, oxidation. Okay? Gain electrons is reduction. Now, I know this doesn't seem to make sense, but we have to first think that the charge was initially 0 or neutral. When we increase the oxidation state, we are increasing its positivity. We are increasing its oxidation value, oxidation state. When we are taking electrons on, we're reducing that. We are making it more negative. Okay, so Leo Ger. We want to think loss of electrons is oxidation, gain electrons is reduction, redox. And let me actually just remove that I, so it's easier to use that for the redox. Okay, so this I help you guys kind of keep that more straight. Now, what's going to happen is these electrons that I'm shuffling about, if I'm taking an electron off, and I've got this electron right here, this electron that I have right here that I've lost, this electron is a packet of energy. Electrons are energy. As I always tell my class, if you don't believe me that electrons are energy, stick your finger in a light socket. Well, don't do that because you'll get electrocuted, you'll get shocked. But the flow of electrons, of electricity that powers our devices, that is electrons. The flow of electrons. Electrons are energy. We can harvest this. So remember that electrons, this is crucial to understanding everything, equals energy. And if we understand that, it's going to be a lot easier to grasp what we're doing. So, with that simple thing out of the way, how do we get these electrons? How are they grabbed? Well, we have to carry them. We have to take them off those carbon-hydrogen bonds and give them to something. We use what is called electron carriers. Now, electron carriers, there are many of them out there that we have. The ones I'm going to talk about with electron carriers are NAD plus and FADH and FAD. Sorry, NAD plus and FAD. We'll turn them into NADH and FADH2s. Now, what's going to happen is these guys, they're bonds, chemical bonds in the glucose, and we can steal these electrons away. Now, let's just kind of think about electrons being energy. Now, as we go through it, I want to give kind of an analogy. Now, if you've ever played poker in your life, maybe you played it on Red Dead Redemption, or you played, electronic poker. You played poker with somebody in real life. When I was in college, me and some of my friends, we would get together some of the weekends. We played poker. We played like Texas Hold'em or something like that. But we didn't play for money. We just played for chips. Now, imagine if I went to Las Vegas. Here, I live in America. So in America, one of the places you go to gamble is Las Vegas. And you go to a casino. And you give your money, and they give you some chips. And you can go to the poker table and buy in. And then if you win, they're going to give you more chips. And you could take these chips, and you could cash them in. And they'll give you money for those chips. And that's what our electrons are going to be like. Our electrons are going to be cashed in for, they're going to be like, it's like our money, and we can cash these in for something else. So like we're playing for chips. You're going to take chips and convert that to money. So our chips, our electron carriers are going to be like our poker chips. Some poker chips are worth more than others, okay? And this is kind of how I like to describe it, is you can cash them in for certain amounts of money. So our electrons have energy. Our electron carriers will hold that energy and transfer it. Now, let's say, if you will, that we have a friend of ours, and this friend, one of the things I found, because like I said, I live in America, but one of the things that happens because I own a pickup truck is if friends of mine need to move, let's say they live in an apartment building in town near, and they live in this apartment complex here, and they said, well, buddy, I need your help. I'm moving. And they're like, I'm moving to a house. I'm moving into a house on the other side of town now, and I need your help to move into my house. And as we always say, friends, they're friends, you can call them, they're going to help you move. So let's say now that they want me to help them move into a house that they now are going to be living in. So they found out that I owned a pickup truck. So they say, can you bring your pickup truck over? And I'm like, okay, I will. And with my pickup truck, I come over, and it is a red truck. I have some of my red pickup. And they're going to tell me, hey, can you take, and we're going to fill up your truck from my apartment. You're going to drive that over here, and you're going to drop it off at my new place, my house. So when we do that, this is kind of what my electron carriers are like. They're carrying electrons. Like I said, now I also gave the poker chip analogy to kind of help us understand what's going on. Now let's use one that is used in all the processes. The one that we have is called NAD+. Let me write that correctly there. Okay. NAD+. It's called nicotinamide adenine dinucleotide. It's derivative of niacin. What we're going to do with this thing is when I fill it up, it was NAD+, but when I go take this guy here, okay? And let's say my friend, I help him move out of his apartment and some of his stuff. And one of the things that he's going to have is 2 electrons and 2 hydrogen ions. And then, so what we do is we turn this NAD plus, because now when I load his stuff into my truck and it's now heavy with all this stuff that I've loaded into it, it is now become NADH, because now it's heavy with his stuff. And so what we then do is when I drive it across, let's say for some reason though, when I'm moving him, one of his hydrogen ions blows out the back. So now when I unload it, I'm going to come here and I'm going to have 2 electrons and only 1 hydrogen ion, and I'm going to deliver it to his new house. And I'm going to go back to being NAD+, okay? Because I've unloaded it. Now what we have is the oxidized state is NAD+. The reduced state is NADH. Now what can happen is, maybe I didn't get everything in the first load. So what I can do is go back and do it again and just keep going till we move him from his apartment to his house, okay? And our electron carriers kind of act like pickup trucks or poker chips, where they carry a value, a certain amount of electrons, and they can then transfer them somewhere else, kind of like how my truck or a poker chip can act. And that's 2 common analogies I use in my class to help my students understand the electron carriers and their role in cellular respiration. Now, who is it that loaded the truck? The truck was loaded by a person here, an enzyme called dehydrogenases. Dehydrogenases are the ones that are going to load our truck. They're going to, dehydrogenase is going to remove the hydrogens with its electrons and help loading it. There's many dehydrogenases out there. For example, alcohol dehydrogenase, aldehyde dehydrogenase, which are used to actually break down ethanol during ethanol metabolism if you drink alcohol. So these dehydrogenases are the ones who are loading this pickup truck, and then as I go back and forth, I load it up, I drive it to his house, I drop it off, I come back, get a new load, and I can keep doing this repeatedly, alternating between NAD+, NADH, back to NAD+, back to NADH, and just keep redoing this, and that's what these electron carriers do, okay? Now, what we're going to do is discuss the last thing is just this big overview of what's going on, the metabolic pathways, some very basics about them, and overall where they occur. Now, I will talk about them more in detail in my next 3 videos. But what we're going to do to understand the 4 metabolic pathways is to discuss these pathways. Now, a metabolic pathway, remember, is just, let me give a very quick definition. A metabolic pathway is a chemical process that starts with a single reactant and then undergoes a series of stepwise chemical reactions where each reaction has its own enzyme and then produces a product unique to that pathway. Now, let's say I've got a cell. And in this cell, we have a cell here. Now, let's say this cell that we have glucose outside that cell, and this glucose gets brought in. Now, glucose, as you guys may know, is being brought into the cell here. This fluid inside the cell, this is the cytosol. And this cytosol, glucose is a 1, 2, 3, 4, 5, 6 carbon sugar. This is a 6 carbon compound called glucose. Now, basically what happens in the cytosol is this 6-carbon glucose here in the cytosol is going to be broken down into 2 3-carbon fragments. Now, if you haven't noticed that what I'm doing here is the dots are representing the carbons, so I'm just drawing carbon skeletons here. When we do this, we get 2 3-carbon compounds called pyruvate. These 2 3-carbon pyruvates, when you produce it, also gets us 2 net ATPs. Now, I will talk about this more in detail later. Overall, this process that occurs in the cytosol is called glycolysis. Okay? So what we're going to do is we're going to be acting like the cell. So with glycolysis here being done, we have 2 3-carbon pyruvates. Now what I want to do is let's take a look. There is an organelle that the cell has. Especially cells who make a lot of energy have this organelle. Lots of them, anyway, a great number of them. And this organelle here is called the mitochondria. Now, most of the chemical reactions of the cellular respiration process occur in conjunction with our mitochondria. Now, let's do a very quick overview of the basics of the mitochondria structure. Remind ourselves that the mitochondria has an outer membrane. There's an outer membrane. There is an inner membrane. There is an intermembrane space. And there is a matrix. Now, also just remind yourselves that the folds of these inner membrane are referred to as the cristae. Now, I'll talk more about that later on in another video, of course. So what we're going to do, let's take one and let's follow this pyruvate. And we're going to bring this pyruvate in, and one of the things that's going to happen is this pyruvate, there will be some CO2 given off by this chemical reaction, and there will be a process that occurs that makes something called acetyl coenzyme A. This chemical reaction here, this metabolic pathway, is called the intermediate reaction. And this is what, and I'm going to abbreviate RXN for reaction, the intermediate reaction is what links glycolysis to the next one. So acetyl coenzyme A is produced here. Now, in the matrix of the mitochondria, there is a very crucial chemical process that occurs. And it occurs 2 times. This is going to take the acetyl-CoA and complete its breakdown, and it's going to be called the citric acid cycle. Citric acid cycle, and it happens 2 times. Now, it does produce some CO2 as well. It also produces 2 molecules of ATP, as well as it produces several electron carriers. Now, right now, I am not mentioning the electron carriers. I will do that in another video where each metabolic pathway gets its own. Now, glycolysis, basically what I'm going to do is a video focusing on glycolysis. Then I will do the intermediate reaction combined with citric acid cycle, and combine those 2 together. okay, in a separate video. But what I want to do is really focus mostly on this overall process of generally what goes on. Now, lastly, what we're going to see, and this is where it always gets kind of tough to be able to write, is on the inner membrane and the cristae, there are all these proteins embedded on it. And these proteins here combine together to produce where a pathway where all the stuff, everything else that is made by the citric acid cycle will go here. And this is a process, and we'll talk more about it in detail later, called oxidative phosphorylation. And it's very hard to write that in this space that I have, oxidative phosphorylation. Now, what we will do is for glycolysis, intermediate and citric acid cycle, which I will combine together in another video, and oxidative phosphorylation. So we will do a video on glycolysis, a video where we combine the intermediate reaction and the citric acid cycle and oxidative phosphorylation in episodes 2, 3, and 4 of this 4-part series. But it's very important that we kind of grasp the overall basics of where everything is, kind of what's going on and what's happening. The big things in this video that I'm focusing on is what catabolic reactions are, what the equation is, and what redox is, and then the electron carriers and the roles of dehydrogenases. Lastly, I always like to go over an overview because this is kind of what is going to be the foundation for the next 3 videos to come. Guys, I hope you found this one useful and helpful into understanding the processes. I will go into more detail for each metabolic pathway, and which will be useful for a foundation for a variety of courses. So guys, thank you so much. As always, again, thank you for watching. Don't forget to comment, like, and subscribe. Let me know what you would like to see me do in future videos. But as always, just thank you for watching. Have a great day.